So yes, we are INBRAIN. We use graphene to decode neural signals into breakthrough medical solutions. We were founded in 2020 because of the vehicle that the European Commission put, the 1 billion to bring graphene to market. Thanks to that, we were able to mature the semiconductors manufacturing technology that we have and raise 15.8 million with investors from Germany and Spain. Additionally, we got a European Pathfinder of 5.2 million that allowed us to go from a 5-people company to a 48-people company with more than 30% of women. We also managed to secure a collaboration with Merck, and INBRAIN does central nervous system, whereas we created this vehicle called INNERVIA for the collaboration with Merck, which focus on peripheral nervous system. So our objective in the future is actually to link both central and peripheral nervous system, because at the end we have one nervous system. On the right-hand side, you have a human brain, nearly 100 billion neurons. On the left side, you have a zebra fish brain of 100,000 neurons. This is how much we know today about the brain, right? With the current tools, with platinum and iridium, this is how much we have learned about the brain. But we need more, because obviously we don't know how the brain works. We cannot fix it. And 1 out of 3 people have a neurological related disease. 30% are refractory to medical treatment, and this generates a huge cost for the society. Now, we need more tools, and every leap in humanity has been linked to a new material, from a Stone Age to Silicon Age, and now in neurotechnology, we need to go beyond. So graphene gives us that opportunity to be the ideal candidate for neurotech revolution. It is a Nobel Prize winner material. It is the thinnest material known to man at an atom thick, yet 200 times stronger than steel, flexible, biocompatible, and with very unique conductive properties. We need it because what we have today is not enough. These are platforms that have been in the market for more than 70 years. Platinum and iridium cannot be miniaturized. And if we want to really reach the broad spectrum of all that potential, we need to go beyond and make sure that we can really convince patients that they need this therapy and not reject it in 50% of their cases, but also understand the biomarkers that actually they are there but we cannot see, and also connect to the environment of the patient to really create personalized therapies. That's exactly what graphene can do. So we can miniaturize, we can read with much higher density and resolution and uncover biomarkers that we have not seen before, and we can couple with modern electronics and bring external sensors and the environment of the data, the environment of the patient, the data of the environment of the patient to actually create that closed loop and that personalization. So combining with all this real life environment patient data, in our case, the strategy is to start from the already reimbursed, let's say, most known BCI in the market, which is DBS. Brain-computer interface, it's a great potential, but there's less than 30 implants worldwide. So start from there and then climb the penetration of central nervous system, and then finally reach the peripheral nervous system and combine the neuroelectronics with the bioelectronics market, making a 25 billion opportunity. How we're going to do it? For the last decades, the brain has been decoded and monitored nuclei by nuclei. We will never get there if we continue like that. We go pathway by pathway, network by network. The first opportunity is Parkinson's disease. And what we do is look at the nigrous striatal pathway, where we actually place an interface on the cortex, an interface on the subthalamic nucleus, and look at the deviations of biomarkers that are pathological, record them, correct them, and make sure that that patient is the highest percentage on time and regaining the quality of life. At the end, the body of the neurons are deep in the brain, but everything converges in the cortex, creating these unique pathways for motor, for psych, and so on. This is exactly what the technology is going to do, and this is exactly what we are building. We have a cortical brain-computer interface module that can go to super ultra high density, up to 1024 contacts, just like Elon Musk. And we have a subcortical module, and everything gets together into a powerful, intelligent chip that is head-mounted with high broad connectivity to the outside world and to a real-life data analysis and processing. I'm not talking about the Inervia here. I said this is the Merck collaboration. It's kind of confidential as well, but at the end, this is the connection we attempt to do very much in the future. Now, this is double-clicking just on the interfacing and leaving aside the chip. This is how it compares to the standard of care. So here you have current technology, Boston, Medtronic, Abbott, and these are the sizes that we have today. This is the lead in the cortex that you will get if you have a brain tumor resection, and this is what you will get operated if you will have Parkinson's disease. Now, this is how we compare, 10 times smaller than the standard of care. Now, you need these because in the same craniotomy where we are going to put 2 contacts, we put 60 contacts as submillimetric size and density. So what graphene gives us here is 2 important features. One, we can miniaturize, as we said. Graphene can inject 200 times more charge density at 10 times lower impedance, so battery management and miniaturization is key. And then we can see 10 times better those biomarkers, so we can detect them and act upon them. We have performed studies in small and large animals. This is the sheep study that we did, feasibility, and also completed GLP. And what you see here is the metal concurrence that we have today, and what you read. So with 2 contacts, which is the maximum that you can fit at that size in the brain, that's how much you read. First, it's not a very good signal. Second, what information do you get out of that signal, right? Same for Parkinson's and so on. The time times better visibility that you get with INBRAIN actually allow us not only to see and react, but also to understand what's going on there. This is the 60 contact interface that we put into the somatosensory cortex of a sheep. And then we touch the tongue on that sheep. And you can see that the darker the color, the more it corresponds to the tongue, right? So you actually understand what's going on there versus 2 lines that don't tell you much. One neurosurgeon in in Barcelona actually told me, this is like wanted to go to Seville and the GPS tells you you are in Europe, right? Now, we have done that in with other evoked potentials, so upper lip, neck, and again, we can see exactly what's going on. If this would be a robotic arm, we will know exactly which finger do we have to connect to actually have fine movement. And again, for our acute cortical applications, if this was a tumor resection procedure, you can safely and precisely remove the tumor without affecting functional areas. I don't know what you prefer if you have a tumor on the brain. Now, the other thing is that we can see things others cannot see. So here you see the contact of the 5 millimeter square, and then you have the comparison with the 1 millimeter square of graphene. Now, everybody wants to close the loop with beta bands. Why? Because it's the only thing metals see. Metals see, platinum and iridium see beta band very clearly, but then if you want to actually close the loop significantly for patients, there's a lot more information we are not seeing. This is like having a very old microscope, right, on the metal side, and then suddenly having a good microscope that shows you what is there so you can create the antibiotic for that bacteria, let's say. And then if you go to the 25 micrometers, which are almost invisible, you start seeing also other biomarkers that are extremely relevant, like high-frequency oscillations are extremely relevant to closing the loop. We have done also subcortical investigations. We can see in Parkinsonian rats deep in the brain from the cortex to the STN single-unit activity and modulation of the beta band with no problem. And of course, with all this high density and resolution, we need a very strong data backbone to actually do the signal processing live, but also to create predictions both to help physicians and patients and empower the healthcare system. We have, in a year, because we got Series A in March, we have done a lot of achievements. We are preparing towards our cortical first in human. We have clear biocom, GLP safety study, subcortical testing, and we still have 1 more year of cash. So we know, we are very confident that we are going to reach our milestone to then go for the 55 million Series B. And the most important is the team, people that have done it again and again. We come from Medtronic, Philips, Sapiens that was acquired by Medtronic, and finally Onward. And all these people came to INBRAIN to realize this purpose and this mission. Finally, we have an amazing global clinical board, and we just created a vision board where we just confirmed the winner, the Nobel Prize Graphene winner, joining us to keep on looking at what is next after graphene and what graphene can give us at the highest potential, and also David Eagleman, which is a neuroscientist at Stanford and an author there. So we are bringing today visibility. We are scientists and humanity lovers. We are going to make this happen, and we need more visionary investors that joined us in our board with the right personalities and with the right constructive mentality to drive and pave the way forward revolutionary neurotech. Thank you so much for listening to me today.